Good morning, everyone. Welcome back. My name is André Brodkorb. I'm from the Teagasc Food Research Center in Moore Park, and I'm actually at home, surrounded by my two proud parents that I haven't seen in 2 years. The webinar is supported by Myrne Egan, who is working in the background. And after the excitement of last month's or May's international conference, we are finally back with our monthly INFOGEST webinar. And today's webinar is organized by the INFOGEST Working Group 4. So they organize it. So without any delay, I will hand over to Frédéric Carriére, Myriam Grundy, and Anabel Mulet-Cabero. And Sophie, of course, our guest speaker.
Thank you, Andre. I would like to thank all the attendees for joining this webinar, and also I would like to thank Andre, Marianne, and Teagasc to make this webinar series possible. I would like to start by sharing some, So I would like to start by saying a couple of words about the Working Group 4. We are a group of around 14 members. The group co-leaders are Myriam Grundy, Frédéric Carriére, and myself. We have a number of objectives, which are mainly to obtain a reliable source of gastric lipase, to find appropriate gastric lipase inhibitors, and optimize the activity measurements for gastric and pancreatic lipases. And Myriam will share some news about that later on. Also, to update and validate the INFOGEST protocol based on the data obtained. And we are also interested in other relevant enzymes such as sterol esterases. So the topic of today's webinar is lipid digestion in early life, which I think is of high interest within the INFOGEST community. This webinar is being recorded and will be uploaded into the INFOGEST YouTube channel, and it also is also streaming live on YouTube. So feel free to make any comments and ask questions throughout the webinar using the Q&A function, which should be at the bottom of your screen. So it is a great pleasure to introduce the speaker of today, Sophie Gallier. Sophie currently works as clinical research manager in the Dairy Goat Co-operative in New Zealand. During her career, she had a great exposure both in academia, working in universities such as California and Massey, and industry where she worked in nutrition research and Frontera. Sophie has a strong background in maternal and infant nutrition, food structure, and digestion of lipids. So now we will play the presentation of Sophie, and I hope you enjoy it.
I would like to thank INFOGEST and the organizers for the invitation to talk today about lipid digestion in early life. As we have greater knowledge of the role of human milk lipids in early life, we now understand that lipids are more than just a source of energy to support infant growth, but that they also contribute to the development of key functions such as cognitive development and immune maturation. A key aspect for the delivery of bioactive lipids is the way they are digested and absorbed in the gastrointestinal tract. Firstly, I want to mention that I'm employed by Dairy Goat Co-operative, an infant formula manufacturer based in New Zealand. As a formula manufacturer, we support the WHO code of marketing of breast milk substitutes and acknowledge that breast milk is the best source of nutrition for infants. Throughout this presentation, we will cover some key aspects of the infant gastrointestinal tract and some in vitro and in vivo models used to understand lipid digestion in infants. Then I will briefly review the lipids in breast milk and infant formula with a focus on the lipid structure. And lastly, I will cover some points about lipid metabolism, brain development, and immune system maturation. I will not go into too much detail on the general topic of lipid digestion, as it has already been covered in the excellent presentation from Claire Beaulieu for the first INFOGEST webinar. So looking at the overall digestive process in infants, there is no oral digestion, whether the infant is fed at the breast or with a bottle. Lipid digestion mainly takes place in the stomach and the small intestine. Just after birth, fecal fat excretion can be up to 10% of ingested fat and decreases to less than 3% after a few weeks. Therefore, some lipids may reach the large intestine where they can be utilized by bacteria and contribute to the maturation of the gut microbiota. There is growing evidence that infants are not born sterile and that colonization starts in utero. Some of the key factors affecting the maturation of the microbiota in infancy are the mode of delivery, whether born vaginally or by C-section, the feeding mode, whether breastfed or formula-fed, the home environment, for example, the presence of pets in the household, and the use of antibiotics, which lead to gut dysbiosis. Infants are not mini adults. The infant digestive system is immature at birth. Presented here are some key differences between the infant and adult gastric systems. Infants have a higher gastric pH in the fasted state, but also during the course of digestion. This is due to limited gastric juice secretion. The infant stomach has lower enzymatic activity, in particular for pepsin, which affects protein digestion. Gastric motility is also lower, and therefore it affects mixing of the chyme with digestive juices. The stomach capacity is also lower, hence a higher number of feeds throughout the day. Infants are breastfed between 8 to 12 times per day and night, and only small volumes at a time, increasing with age. As shown on the left, in an older study in infants in the second week postnatally, the infant's gastric pH increased rapidly after a feed and slowly decreased and returned to fasted conditions after 3 hours. The gastric pH remained above 4 for 2 hours. These pH conditions limit the activity of pepsin but are more optimal for the activity of gastric lipase. On the right-hand side of the slide, Bio et al. Measured gastric emptying in infants in the first year of life by labeling breast milk and formula with a tracer and measuring residual gastric radioactivity. The study demonstrated that gastric emptying was not affected by age when infants were fed the same way dominant formula. In the same study by Bio et al. On the left, they also looked at the gastric emptying based on the type of milk. A consensus with other studies is that human milk empties faster than most formulas, and this relates to the type and content of proteins, with human milk being a more acid-stable emulsion and having more soft or yogurt-like curd properties under gastric conditions than cow's milk-based formula, forming a harder curd. Here in the center of the slide, the study by Yu et al. Using ultrasound measurement in preterm infants up to 3 weeks of age showed similarly a faster gastric emptying for human milk. Overall, whey-dominant formula emptied faster than casein-dominant formula. To note that this is true when comparing formulas based on cow's milk, but differences may not be present when comparing formulas based on milk from other species, such as goat milk, as its curd properties under stomach conditions are closer to that of human milk. Formulas with extensive or partially hydrolyzed proteins tend to empty faster than formulas with intact proteins. Again, this is linked to the properties of the curd. The influence of protein content on gastric emptying is demonstrated here with follow-on formula and cow's milk, both having a higher protein content than human milk and infant formulas, and emptying more slowly from the infant stomach. While gastric pH decreases slowly after a feed, gastric emptying is quite rapid, as shown here with the T-half graphs representing the time it takes for 50% of the feed to empty out from the stomach. The short gastric residence time in combination with pepsin being optimally active at pH around 2 can limit significantly protein digestion in the infant stomach. While gastric motility is low in infants, peristaltic contractions drive the mixing of the chyme with gastric juice and movement of the chyme down the stomach to be emptied out in the duodenum. Some factors affect gastric emptying and can result in the gastrointestinal discomfort, such as regurgitation. Milk will curdle under gastric conditions and therefore lead to a solid phase composed of a casein curd and entrapped lipids, and a liquid phase with soluble proteins and lactose. The liquid phase empties first. The phase separation is driven by the curd properties, which relates to the type and content of proteins. To be emptied out from the stomach, the curd particle size needs to be reduced down to less than 1 or 2 millimeters. The size of the particles is controlled by gastric mixing, pH, and digestion of nutrients. And lastly, the caloric value and volume of the feed will also impact gastric emptying in infants. Gastrointestinal enzyme secretion starts prenatally. Enzymes involved in lipid digestion are gastric lipase, secreted in the stomach, and pancreatic lipase-related proteins 1 and 2, bile-salt-stimulated lipase, and phospholipase A2 released in the small intestine. To note that human milk contains an endogenous bile-salt-stimulated lipase, which can also contribute to gastrointestinal lipolysis. The activity of gastric lipase at birth is comparable to that of adults, whereas pancreatic lipase activity is very low at birth and slowly increases in the first months of life. The other lipases secreted in the small intestine play therefore a role to digest lipids in concert with gastric lipase, which remains active under intestinal conditions. In the stomach, gastric lipase can hydrolyze 10 to 30% of triglycerides into free fatty acids and diglycerides. In the small intestine, the gastric lipase continues the digestion of triglycerides in combination with pancreatic lipase, which is SN1 and SN3 specific, and other lipases that have no specificity and can hydrolyze most lipids. The digestion of glycolipids and cholesterol is more complex and to some extent not fully elucidated, but some may reach the large intestine where they may be fermented by gut bacteria. Bioaccessibility of lipids for lipolysis can be affected by several factors. The strength of the protein curd and trapping lipids can limit the diffusion of enzymes. As lipid digestion is an interfacial process, the size of the lipid droplets and the composition of the interface plays a key role. We will touch on those points later in the presentation. While the study of infant lipid digestion is not new, there has been significant increase in the number of publications and improvement of in vitro models replicating more closely the infant digestive system. The most representative and feasible in vitro model is the one developed by the INFOGEST group and published by Mena et al. In 2018. As indicated previously, there is no oral phase. In this static system, the gastric pH is set at 5.3, the gastric residence time is 60 minutes, and gastric lipolysis carried out by a gastric extract. The intestinal pH is set at 6.6, the intestinal residence time is also 60 minutes, and porcine pancreatine supplies the blend of enzymes, and bovine bile is added. A range of in vitro dynamic models have been developed using known and validated in vivo parameters. These systems can simulate gastrointestinal motility and emptying and continuous secretion of gastrointestinal juices. Some of these systems are the DG system developed by INRAE et al., as shown here on the left, and the MIDA, or model of infant digestive apparatus, here on the right, and developed by Heinz and researchers in Italy. Other systems use filtration membranes to simulate the absorption of digested nutrients, such as the TIM Pediatric, an adapted version of the Tiny TIM from TNO, and the simulator of the human intestinal microbial ecosystem, or SHIME, from ProDigest. The SHIME model is also an example of in vitro models using a colonic fermentation unit, using stool samples collected from infants. Human clinical trials are the best approach to study food digestion. However, getting ethical agreement to conduct gastrointestinal digestion experiment on humans is difficult, and even more so on healthy populations like infants. As mentioned earlier, infants are not mini adults and have different digestive patterns to that of adults. In vitro models have significantly progressed, but they still have some limitations. For those reasons, animal models have been widely used. However, many studies have used rodents, despite some significant physiological differences, such as the type of lipases and translation of microbiota results. When focusing on infant digestion, the use of piglets remains the best model to understand the process of infant gastrointestinal digestion. Breast milk is a complex and dynamic source of personalized nutrition for the infant. Focusing on breast milk lipids only, they provide up to 50% of total daily energy for infant growth. The level of fat varies during the feed, with the fat concentration increasing between the full milk, when the infant starts suckling and the hind milk within minutes of feeding. The fat content also varies across lactation and the time of the day. Maternal nutrition has an impact on the composition of breast milk. For example, mothers living in coastal areas and therefore consuming more fish tend to have higher levels of DHA in their milk. Human milk contains a variety of fatty acids, up to 15% of PUFAs, and is a rich source of healthy PUFAs, unlike milk from animal species. But like cow and goat milk, human milk contains fatty acids that are not found in vegetable oils, such as pentadecanoic acid, palmitoleic acid, and heptadecanoic acid. Another key feature of human milk is that it contains 16 to 25% of palmitic acid, of which up to 70% is found in the SN2 position of triglycerides, whereas only 30% of palmitic acid in cow and goat milk are in the SN2 position, and vegetable oils mainly contain palmitic acid in the SN1 and SN3 positions. The SN2 position of palmitic acid in human milk facilitates its digestion and absorption as monoglyceride. Human milk is more than just the sum of its nutrients. Some nutrients are packaged in a way to enhance nutrient digestion, absorption, and delivery within the infant gastrointestinal tract. Commonly to milk from all species, human milk lipids are present as milk fat globules, surrounded by the milk fat globule membrane, or MFGM. The MFGM originates from the unique secretion of lipids in the mammary cells and is made of a monolayer of phospholipids and proteins from the endoplasmic reticulum, a bilayer from the apical plasma membrane of the mammary cell, composed of phospholipids, cholesterol, and proteins, including glycoproteins. The glycan residues on glycolipids and glycoproteins form the glycocalyx. The MFGM stabilizes the triglycerides, protects the globules from lipolysis, but also provides bioactives for cognitive and immune development. Cholesterol is present at similar levels in milk fat or MFGM of human, cow, and goat milk, but is absent in vegetable oils that are used to manufacture infant formula. In addition, the MFGM phospholipid content and distribution is similar in the three milks compared here, with the exception of sphingomyelin, representing a higher percentage of total phospholipid in human milk. Human milk is not always consumed directly at the breast and must therefore be stored under appropriate conditions for later consumption by the infant or at human milk banks. Heat treatment of human milk usually consists of Holder pasteurization, which is at mild temperature but sufficient enough to lead to a change in the interfacial composition of the MFGM with whey proteins interacting with milk fat globule membrane proteins. Other human milk proteins are denatured, such as the bisalt-stimulated lipase, which loses its activity, thereby reducing its ability to digest lipids in the gastrointestinal tract. Human milk is stored in the fridge for a couple of hours or in the freezer for longer storage. Freezing in particular leads to crystallization of the lipids and rupture of the MFGM. Upon thawing, this leads to a restructuring of the fat globules and absorption of milk proteins on the surface of fat globules. Freezing, thawing, and pasteurization increase lipolysis in milk pre-digestion, but did not impact gastric lipolysis in preterm infants, as studied by De Oliveira in 2017. In a 2016 study using a dynamic in vitro system, the same group reported a decrease in gastrointestinal bioaccessibility of some fatty acids due to pasteurization of preterm human milk. Human milk is the gold reference to establish the composition of infant formula. There has been significant improvement in the manufacture and composition of infant formula in the last 100 years or so, but we are still far from being able to replicate the complexity of human milk. Most infant formulas are manufactured as shown here. Bovine skim milk and whey protein powders are mixed together to obtain a 60-40 whey-casein ratio. Then lactose, vitamins, and minerals are added or homogenized with vegetable oils. This results in the formation of stable submicronic oil droplets stabilized by milk proteins. Other ingredients are often added at the dry blending stage. The first infant formulas were made with animal milk. However, to meet infants' needs, vegetable oils need to be added to match the fatty acid composition of human milk. Over time, milk has been fully replaced by vegetable oils and fungal and marine oils to provide healthy PUFAs. However, milk fat is being reconsidered for the manufacture of formula due to the presence of contaminants such as 2 and 3 MCPDs and glycidyl esters in vegetable oils, the environmental impact of palm oil negatively perceived by consumers, and the recognition that milk fat provides MFGM components, SN2 palmitic acid, and cholesterol, whereas vegetable oils contain phytosterols, palmitic acid in the SN1 and 3 position, and no MFGM. There are a few ways to add milk fat using either a similar process to the standard one by adding whole milk, cream, or AMF. The way we manufacture at DGC, we use whole goat milk without adjusting with whey, allowing us to have about 50% of the lipid source as milk fat without the need to add palm oil to supply palmitic acid in the formula. Here we have a brief overview of infant formulas formulated to improve lipid digestion. For preterms, formulas may contain medium-chain triglycerides for faster digestion and absorption in a very immature digestive system. Another type of formula is based on the fact that standard formula may form a harder curd under gastric digestion, trapping lipid droplets and limiting the diffusion of enzymes through the curd, leading to limited hydrolysis and symptoms of discomfort such as speed-ups in regurgitation linked with the slow gastric emptying. Those formulas are hydrolyzed formulas containing pre-digested proteins, goat milk-based formulas with goat milk forming a softer curd than cow's milk under acidic conditions, and more recently some development on formulas based on cow's milk proteins with a lower casein mineralization, also for allowing to form a softer curd. Formulas containing milk fat have an improved lipid composition and triglyceride structure. Similarly, for formulas with OPO from palm oil, modified by enzymatic interesterification to shift palmitic acid from the SN1-3 position to the SN2 position. There are also formulas with no palm oil or no milk fat, so with low levels of palmitic acid. However, it is unclear if there may be negative long-term health outcomes in supplying low levels of palmitic acid in early life. Other developments have been around the lipid structure, recognizing that with the removal of milk fat, the milk fat globule membrane is not present in standard formula, and formula with old droplets have a different structure than human milk fat globules. MFGM-rich ingredients, such as beta serum from AMF processing and MFGM-enriched whey from cheese processing, are being added to formulas. Regulatory requirements are focused on some key aspects of lipid composition, such as providing essential fatty acids at specific ratios, but these requirements do not take into account the triglyceride structure and microstructure specificity of human milk lipids. Vegetable oils commonly used in infant formula provide medium-chain fatty acid, palmitic acid, and long-chain polyunsaturated fatty acids. To supply a more diverse fatty acid profile, including short-chain fatty acids and palmitic acid, some of which in the SN2 position, while limiting the presence of contaminants, a growing number of formulas include milk fat, either from whole milk, cream, or AMF. In the table is an example here of the impact of on the percentage of total palmitic acid and SN2 palmitic acid in infant formula, depending on the source of lipids. Infant formula without palm oil or without milk fat have low levels of both, whereas infant formula with palm oil or PO or milk fat can reach total palmitic acid levels close to that of human milk. However, SN2 palmitic acid levels remain lower than that of human milk, even for infant formula with milk fat or PO. This is from a project I worked on when I was at Danone Nutricia. On the right, this is a schematic representation of the human milk fat globule, which has a diameter between 3 and 5 microns and surrounding by the MFGM. On the left, this represents standard infant formula fat droplets with vegetable oils stabilized by milk proteins. The Nutrius concept in the middle was manufactured using an MFGM-rich ingredient called beta serum and with adapted manufacturing parameters, resulting in a larger droplet size and a surface stabilized by milk proteins, but also MFGM components. Using a two-step semi-dynamic model of in vitro gastrointestinal digestion and measuring free fatty acid analysis with gas chromatography, it was shown that the lipid digestion of nutritious formula was more similar to that of human milk than the standard formula. Different digestion kinetics could lead to different bioavailability of lipids within the gastrointestinal tract. Beaulieu et al. Explored the impact of the size and interfacial composition of lipid droplets on the in vitro gastric digestion of model infant formulas. So M1 is a minimally processed formula from raw milk, minimally heat-treated, skimmed, standardized, and with added whey to reach a 60-40 whey-casein ratio. The M2 formula is an homogenized version of M1, and the M3 formula is a pasteurized version of M2. M1 displayed the lowest degree of hydrolysis, and M3 the highest degree of hydrolysis. The release of free fatty acids was measured with gas chromatography. This profile was marked by high levels of short-chain and medium-chain fatty acids and lower ones in palmitic, myristic, and oleic acids at the beginning of digestion of M2 and M3, whereas the release of short to medium-chain fatty acids was delayed in M1 in relation with its lower initial lipolysis. Armand et al. Studied gastric digestion of lipids in preterm infants fed either human milk or one of two infant formulas. Despite having a larger droplet size and therefore smaller interfacial area for the gastric lipase to anchor at the surface of the droplets, the gastric lipolysis of human milk was significantly faster than that of infant formulas. This highlights the role of the MFGM in facilitating gastric lipolysis. The difference with the study by Beaulieu et al. Is that in this study by Armand et al., the fatty acid composition and protein content varied between human milk and formulas, whereas they were kept constant in the study by Beaulieu et al. The study by Armand et al. Highlighted a concept of lipid microstructuring during digestion. They used transmission electron microscopy to look at the microstructure of milk before and during gastric digestion. After 50 minutes of gastric digestion, water pockets appear between the milk fat globule membrane and triglyceride core of human milk fat globules, whereas in infant formula, the water pockets were found within the triglyceride core of the oil droplets. Lipolytic products, as seen as darker spots, were also found within the core of the droplets. This microstructuring concept has been recently studied further by Professor Boyd and his team at Monash University. They looked at the intestinal digestion of human, bovine, and goat milk and 3 different infant formulas and analyzed the self-assembly of liquid crystalline structures formed during digestion with sacs at the synchrotron in Melbourne. All milk self-assembled into other structures during digestion, with the majority of milks displaying non-lamellar phases. Milks that release mostly long-chain fatty acids predominantly formed a micellar cubic phase during digestion. Other milks release relatively more medium-chain fatty acids and medium-chain monoglycerides and produced a range of ordered liquid crystalline structures, including the micellar cubic phase, the hexagonal phase, and the bicontinuous cubic phase. One infant formula did not form liquid crystalline structures at all as a consequence of differences in fatty acid distributions. The structure from infant formula digestion were closer to that of human milk, and likely due to the differences in PUFAs and LC PUFAs with bovine milk and goat milk. Lastly, we will go over some of the nutritional and physiological functions of lipids in relation to how they are digested. The diversity of milk fatty acids and the specificity of the gastrointestinal enzymes may control the digestion kinetics of milk fat. For example, short-chain fatty acids and medium-chain fatty acids are easily digested and absorbed. Palmitic acid in the SN2 position is released by pancreatic lipase as monoglyceride, which can be easily absorbed. Palmitic acid in the SN1 or 3 position is released as free fatty acids and can form soaps with calcium, which can lead to hard stools, constipation, colicky symptoms, and increased crying in infants. There has been a few clinical trials in infants fed formulas with milk fat or OPO demonstrating improved gastrointestinal outcomes, in particular stool characteristics, compared with infants fed a formula with palm oil. By reducing the formation of sops, this also allows improved calcium absorption and subsequently bone strength. Cholesterol is important in infancy as a precursor of steroid hormones, vitamin D and bile acids, and to regulate cholesterol homeostasis later in life. Cholesterol and bile salts secreted in bile are important for lipid digestion and absorption. Cholesterol and bile salts that are not absorbed can be fermented by gut bacteria in the large intestine. High milk fat levels in formula allows to increase the levels of cholesterol, but also to limit the amount of plant sterols present in vegetable oils. The impact of the presence of plant sterols in infant formula is still unclear. Back to the nutritious concept, the increased size of infant formula droplets and the interfacial coating being closer to the composition of the MFGM have advantages over standard infant formula with regards to lipid metabolism and metabolic programming in early life. In infants, the nutritious concept with a 50-50 blend of milk fat and vegetable oils was shown to support healthy growth, was well tolerated, and resulted in stool characteristics similar to those of breastfed infants. In vivo models, where rodents were fed a control or nutritious formula in early life and then fed a Western-style diet in adulthood, it was demonstrated that the early life consumption of nutritious formula resulted in higher hepatic levels of proteins involved in fatty acid metabolism and oxidation, increased gene and protein expression of mitochondrial oxidative capacity markers in white adipose tissue and skeletal muscles, and reduced excessive body fat accumulation. Milk fat and the MFGM play also a role in brain development and cognition. Milk, and in particular goat milk, is a rich source of medium-chain fatty acids. Medium-chain fatty acids prevent oxidation of PUFAs, but also enhance their bioconversion to healthy PUFAs, leading, for example, to greater accumulation of DHA in the brain. Medium-chain fatty acids are rapidly absorbed in the small intestine, transported to the liver, where they are beta-oxidized to acetyl-CoA. Thus, medium-chain fatty acids can be substrates for ketone bodies, contributing to ketogenesis. The MFGM is a rich source of lipids also found in abundance in the brain, thus can supply building blocks for the synthesis and maintenance of brain cells and neurotransmitters. A few recent studies have demonstrated that supplementation with MFGM in infancy contributes to the development of the brain and cognitive functions in healthy-term infants. Here, an example of a study in Chinese infants fed for 12 months a controlled formula or a formula enriching MFGM and lactoferrin. Infants fed the supplemented formula had higher scores of cognitive, language, and motor development measured with the Bailey-III test at 12 months of age. Similarly, the studies with MFGM supplementation using an MFGM-rich fraction to manufacture infant formula have demonstrated a protective role against infections in infancy. The mechanism of action likely involve antimicrobial properties of milk lipids, lipolytic products with detergent-like properties, and MFGM proteins. MFGM glycosylated proteins and lipids can act as decoy receptors for pathogens and viruses, preventing their adherence to the intestinal wall, but also limiting their growth within the gastrointestinal tract. MFGM sphingolipids have been shown to promote gut maturation. Animal studies and clinical studies with milk fat and/or MFGM have also shown a modulatory effect on the gut microbiota and overall an effect on the gut-brain axis. A few take-home messages to finish. The infant digestive system is immature at birth, and therefore digestion of nutrients is not the same as in adults. Human milk lipid composition and structure is complex and different to that of infant formula. Lipids are more than just a source of fuel in early life and contribute to metabolic programming, brain development, and immune maturation. And adding milk fat to infant formula has advantages over vegetable oil-based infant formulas for nutritional and functional outcomes for the infant. Thank you very much for your attention, and I'll be happy to answer any questions you may have.
Thanks a lot, Sophie, for this very interesting and excellent presentation. We are now moving on to the Q&A session, so I will invite Sophie, Myriam, Frédéric to join and switch on the microphone and camera. So just a reminder that you can submit the questions using the Q&A function at the bottom of your screen. So, Frédéric, would you like to start?
Okay, thank you, Anabel. And thank you, Sophie, for your very nice presentation. That's a pleasure to have you today. So it was a very interesting talk. So I will have first a question about your tries to mimic human fat globules when you were working at Danone. So you mentioned the fact that you try to engineer the monolayer with polar lipids. Are you aware of attempts trying to make a full 3-layer of polar lipids around the lipid droplet in order to be closer to the real fat globule?
From a manufacturing point of view, that's probably still going to be very challenging. Maybe with some microfluidic devices, you could do it in the lab. But even the structure of the MFGM in the MFGM-rich ingredient, it's broken down. Whether you get it from the AMF processing or from the cheese making processing, you do not get the full intact structure. So currently, impossible to do it in the factory, but something that we are still all trying to achieve to be as close as possible to human milk.
I understand. And then another particularity is the presence of medium chain fatty acids in this milk as a human goat from bovine origin. And so I was expecting you told us about the fact that they can be absorbed at the gastric level. Did you investigate that?
No, we didn't do any investigation on that side, and I know it's still unclear whether or not the short chain fatty acids and the medium chain fatty acid can be absorbed in the gastrointestine, well, in the gastric compartment, or whether they absorb directly in the small intestine as soon as they reach the duodenum. We didn't study that at all. Usually in infant formula, one of the main source of medium chain fatty acid is also coconut oil. So there might be a little bit more information around coconut oil absorption, maybe in adult system on that.
It's true there are not so many studies on the absorption of short and medium chain fatty acid at the stomach level, and the one that I know are rather hard, but they were done in the rabbit using a closed pouch in the stomach. And it was done by a veterinarian in France and called Perret, and they have shown that fatty acid could be absorbed at a chain length of C12 in the stomach. So I was much interested in that because due to the specificity of gastric lipase releasing these fatty acids preferentially from the milk, it seems there is a connection between the fact you can release them already in the stomach and the fact that can be already absorbed there. So as you said, there are not too many studies on that right now.
No, and that could be also of interest for preterm formulas where some of them are enriched in MCTs. Okay. So, but no, I'm not aware of any particular studies there.
Okay, thank you, Sophie. So maybe I will let Anabel and Myriam ask other questions, or people attending the. Myriam?
Yes, there are some few questions from the audience. So there is one. Is there a difference in the caloric content between human and cow milk?
There's no difference in energy, and well, for infants 0 to 6 months and even 0 to 12 months of age, cow's milk is not a source of nutrition. Something we need to mention. But if you look at the regulation anyway for infant formula and follow-on formula, there is a limit, a min and a max that we need to achieve for the energy content. So it's between 60 and 70 kilocal per 100 ml. So that's regulated, and that's based on the calorie content of human milk. There's still some differences in some of the micronutrients. So for example, for the protein content, that it's still protein content in infant formula is still a little bit higher than in human milk, but all of that is regulated to try to match the composition of human milk as close as possible.
Okay, thank you.
We have another question for the audience. So how do you study the effect of digestion on milk fat globule membranes? How do you know they are affected by digestion?
So some of the studies have looked at the digestion of human milk, for example. So whether in the gastric phase or in the intestinal phase, and looking at the protein degradation. So proteins can be digested by pepsin chymotrypsin and trypsin. Some of the MFGM glycoproteins are still resistant to digestion, so they've been found in the infant stools, for example. With regards to the MFGM phospholipids, they will be digested by the phospholipase A2 in the small intestine. And so you could look also at the degradation of those. For some of the glycolipids, it's still a little bit unclear how they're digested, for example, gangliosides. There's been some work by Beaulieu et al., and I've done also some work on that when I was in California, extracting some of the MFGM phospholipids and looking at the digestion, the impact of the digestion on the structure of the MFGM phospholipid using some Langmuir monolayers. So that's another way to look at it. Then there is all the MFGM-rich ingredients that are added to infant formula. So the structure is quite different for those ingredients. So the MFGM has been pulled apart, so it's not present at the surface of the milk fat globules. So the digestion of this type of MFGM might be different to the MFGM present at the surface of the globules.
Yeah, okay. Well, maybe another question. Is there a clear difference in digestion pattern between goat milk and bovine milk?
So regarding the digestion of the lipids between the two, there's not really much comparison. There's more work that's been done for the protein digestion, and there are some differences. So if we look at the caseins, there seems to be a faster digestion of the goat milk caseins. And there's been also some work done by Matas et al. On the digestion of infant formula made from goat milk and infant formula made from cow's milk. Both were whey adjusted, and there was also a faster digestion of the whey, the goat milk whey and casein proteins.
Myriam, would you like to ask another?
Yes. So somebody will find your talk excellent, and thank you for that. So you show the effect of MFGM on brain development in the infant, in a study done in Asia. Is there any follow-up being done showing which component of MFGM is the most bioactive regarding brain development?
Yes, so a lot of the work in this space has been sponsored by industry, and so some companies are focusing on one particular aspect rather than another one. So overall, it's probably for brain development, it's more around the MFGM lipids rather than the proteins playing a role. And so at Fonterra, we focus very much on gangliosides, so the companies are focused more on the presence of sphingomyelin, also in the MFGM. What is still unclear is the fact that the MFGM that we have in infant formula has been processed, and whether or not there's a loss of bioactivity compared to the MFGM in human milk. Another aspect is that there's differences in ratio of the phospholipids between the MFGM in human milk and the MFGM that you can get from cow's milk, for example, whether or not that has also an importance. So it's still very much unclear. I think that the best answer at this stage is probably a combination of several lipids that are present in the MFGM that play a role on brain development.
Okay, thank you. Do we have more time for questions?
Yeah, we have 5 minutes more. There is another question about the percentage of fat absorbed. They don't specify in which part of the gastrointestinal tract, so maybe you can just say again, because I think you said in the talk, how much percentage of fat is absorbed in the gastric part, in the intestine.
So in the gastric part is very close to what we see in adults, so 10 to 30%. I think from a review by Beaulieu et al. In the small intestine, that was 56% of the fat that was absorbed. overall, fat digestion is quite efficient in healthy term infants. Usually we see between 95 and 98% that's absorbed. It's more in that really those first few weeks of life where the system is quite immature. So there's been some reports of up to 10% of the fat that can be excreted in the stools.
Good. Frédéric, do you have any other question?
Yes, maybe I can ask a question about the gastric pH and the contribution of enzymes in the stomach. So what about the salivary amylase? Even if you are in a session dedicated to lipids today, I am interested by what would happen to carbohydrates in the stomachs in the infants.
So if you look at the composition of human milk and the infant formula, the main carbohydrate is lactose. Mm-hmm. And then you've got the human milk oligosaccharides. So human milk oligosaccharides usually are not digested and they will reach the large intestine where they are fermented by the bacteria. For the lactose, it's digested by the lactase at the brush border membrane. So the amylase, from what I understand, is that the secretions takes off once the infant is switched to solid food. Okay. So more towards the age between 4 and 6 months of age. So that's where the amylase may play a role and the secretion might be more important.
I was asking this question because of the recent work of Daniela Freitas, who showed the role of salivary amylase already in the stomach. And this role is at a pH that is, as you said, for gastric lipase is quite not maybe optimum, but relevant. Whereas you said that it was not so good for pepsin. And today I'm wondering whether in fact we are not wrong about the correct pH we should use for looking at digestion in the stomach and the role of the various enzymes. And maybe both gastric lipase, salivary amylase, and pepsins, they work at a similar pH, which is close to 4 and 5. And we can see today for several experiments that even at pH 5, pepsin has still some high activity. So I was wondering whether you explore also the digestion of protein from the milk at a higher pH than 2 or 3.
I think we had done some work up to pH 5, and we still saw some digestion, but it's fairly limited. But yeah, that's maybe something to explore a bit further.
Okay, thank you, Sophie.
Thanks.
Okay.
Can I ask a question?
Yeah, sure.
I have a question for the whole panel, not just Sophie. So you mentioned earlier that some of the fat is absorbed in the stomach. Why is that? Why is there any absorption in the stomach at all?
I think my understanding of that is that it's a fast way of getting energy, a bit as you will get with the carbohydrates. So that's what I understood from the literature. And I said to Sophie, and they are not the literature about this absorption in the stomach is not huge. There are this work from Perret in France, who's veterinarian who studied that in the 70s and 80s, I think. And more recently, there was another group in France at Rennes, Vincent Rioux, who studied the absorption in the rat. So this is also documented in rats that you can have up to C12, I think, C8 for sure. And the reason why it was reinvestigated recently is that maybe the C8 that is absorbed at the gastric level could be used for the octanoylation of the ghrelin hormone, which is important for food intake. It was not fully demonstrated, but these are the questions that are currently investigated by some labs.
And that remains throughout your life. It's not just in infants, yeah?
I'm not sure this has been investigated in adults. So.
Fascinating. Yeah, very good.
Well, we don't have more time for questions, I'm afraid. So I would like to thank Sophie again for the presentation and for your time. Thank you very much.
Thank you.
And I will hand over to Myriam that has some news to share.
Yes, so I'm going to share my screen. I hope it's going to work.
Geraldine,
Up.
to Canada, please.
So can you see my screen?
Yes.
Okay, perfect. So I just wanted to share with you actually our good news, I think, for the achievement of our working group activities, the first one, I hope. So we recently, so it was in May, I think, he was online. So we published a work on lipase activity measurement from gastric and pancreatic lipase. So it's been published in the Journal of Functional Food. As you can see, a lot of people took part in this work. So I think we've got 46 authors on that paper. So I thanks again all the people involved in that work. So what we show actually, it's lipase activity have to be determined prior to in vitro digestion study. So in particular, if you're looking at lipid digestion, and the pH stat method give quite good reproducible data. And this we show within individual laboratory. And the pH stat equipment parameter needs to be considered. So depending on which pH stat you use, or which kind of vessel and which kind of steering material you've got, this can affect actually the accuracy and reproducibility of your measurement and activity assessment. In particular, the reaction vessel shape significantly impacts the reproducibility of the result. And we also looked at one inhibitor in particular, boronic acid, and is quite effective for the intestinal phase, but not so much for the gastric one. So if I show you quickly some of the results that we published in that paper. So on the left here, you can see a schematic of pH stats. So with a pH electrode, they measure the pH. You've got your stirrer. It could be a propeller like that, or little stirrer. And you're having the NaOH in general 0.1 N in concentration to keep the pH constant while the free fatty acids are released. And you will see, so the diagram on the top right, then when you double your concentration, you should see a double in activity. And that activity is increasing linearly, in particular the 5-10 first minutes of your measurement after you add a little lag time. So a few seconds it takes in general. And you will see from the diagram on the bottom, right, that you need to be in a range, in the linear range of your enzyme concentration when you measure your activity. Otherwise, you will see, you will underestimate the activity, as it is the case since it's a diagram with a not marked pancreatin, for instance. So you have to be really careful and use different enzyme concentration to make sure that you're not underestimating your activity. Okay, so that's it from me, and you've got your details if you want to contact us. Thank you.
Yeah, so now, André, over to you.
Very good. So first of all, I would like to thank the panelists, the three panelists and the presenter. I think was a really good talk. I really enjoyed it. Good overview. I think we have a lot of attendance from the non-lipid world. So it was a great overview. Thank you again for staying up late. It's early for us, it's late for you. Okay, thanks again. So I just have three more slides. Give me a second. Here we go. Can you see my slides?
Yes.
Yeah, very good. Just a word or two about the INFOGEST network, because we have a few new attendants. Overall, we have about 60 attendants, a few people on YouTube as well. So the chair is Didier Dupont, and Alan Mackey is vice chair. We have 6 working groups, and you see them, spanning different areas of food digestion. And I really encourage the working group leaders to come up with new ideas for webinars. And I really like to thank the 3 working group leaders today. Here you go. Our next webinar is after summer, of course. Every first Wednesday of each month at 2 o'clock Dublin or UK time. It is a joint webinar with Cost Action UNGAP on the food-drug interaction. We have 1 confirmed speaker, Bart Hens, and the second one is nearly confirmed, so it will be a good change. We streamed today's webinar live on YouTube, and subsequently we leave all the webinars up on our YouTube channel. If you just Google in vitro food digestion in YouTube or scan this QR code, it's easy enough to get there. So that's my last slide. Oh no, one more. Just watch that space. We have a LinkedIn group. So if you go to LinkedIn, INFOGEST, type in INFOGEST. We have about 800 followers. You can post yourself. So if you want to post a link to an article, all you have to do is @INFOGEST, and 800 people will actually see this post. Alternatively, you can join the INFOGEST network by writing an email to Nathalie Demar or Didier Dupont. So until then, hope to see you again on the first Wednesday in September, 1st of September, 2 p.m. Slán, and goodbye.